So this is basically a talk I'm giving on behalf of a bunch of people, which some of which are in the room, including Paul Yamina. And Paul and I head the Australian Grains Pest Innovation Program, or AGPIP for short. And that's based on developing a whole lot of blue sky stuff, as well as doing some very basic research on insecticide resistance. So we sort of cover a bunch of bases. So for instance, one of the things we're doing, blue sky, is creating aphids that can't actually transmit virus anymore. And we make a lot of progress in that area. So it's very nice to be here, and I will go fairly quickly because I realise that we're running a bit behind time, or at least I'll try. And this was sort of a warning paper that Paul and I and a bunch of other people came up with a few years ago, just to remind people that resistance is escalating in insects and mites, and this is a problem we face. And we've heard some stories already this morning about blue-green aphid. It's obviously a new one that's come off, and Everett will talk about that later. But there's a whole lot of stuff that is going to be happening in the future, which is going to basically complicate our resistance issues, unfortunately. We can think of a number of those. So, of course, what you want to do is you want to try and come up with a holy grail that will help predict what's going to happen in the future. So you really want to sort of try and develop with a crystal ball. And then, of course, what you want to do is to try and manage what's happening, try and reduce the resistance. And, of course, what you didn't want to do is to say, we give up, right, let's move on to the next thing. So, I mean, that's what resistance is all about in insects and mite pests. So how far can we get? What can we do? How do we actually solve this sort of issue? So the Holy Grail is one that we chase, and of course we chase it as researchers, and of course that eventually hopefully feeds back to growers and keeps the industry viable. So as a consequence of this sort of work, what you want to do eventually, of course, is that when resistance develops, you want to develop resistance management programs, and we've developed a number of those over the years, and here's one that's been around for a while on red-legged earthworm that we heard about this morning. But we've just updated this year, so we keep updating these things. But of course, you've got to ask yourself, well, how effective are these? We heard this morning about the competing interests in terms of particular high valuable crops where you keep growing the same thing. It's going to happen, you're going to get a problem, and how do you actually do that? So we develop these, we come up with recommendations, but are these recommendations ever useful? And that's a challenge that we face. That might be a good topic for discussion at some point in time. So there's a lot of science that goes into these sorts of things. We make sure they're scientifically rigorous. And of course, as the science comes in, and as the data comes in, we can improve these sorts of things and make them better. But at the same time, there's still the issue of how you actually adopt these and implement these programs. So one of the things that we started doing this year, we started this in AGPIP a few years ago, but we've really started publishing on this stuff this year, is to tackle the issue of, can we predict resistance before it actually happens? Can we get a handle on what's going to happen? Can we get a handle on what you're facing in the future? Let's look at the crystal ball and have a look at that. So basically, the problem that we faced when we started doing this exercise, and this is a paper that James Mayno led, that was published earlier this year, was how do you actually measure the issue of how much pesticide is going on? What do we know about the selection pressure that is driving resistance? So in a sense, insect and mite pests are no different to what Chris was talking about. You've got issues where basically the more you select, the more likely you are to get resistance. And this has turned out to be quite an important issue. And we don't have good data in Australia, so we developed a proxy, if you like, which is the number of products that are being registered, and that seems to give a bit of an indication of pesticide pressures on insects and mites, at least in Australia and the US. So that was quite an important breakthrough for us. So what we then did was we didn't say, well, let's take that particular measure and compare it to other measures that might help us predict what's going to happen in the future. Can we come up with predictive measures that sort of include these sorts of parameters? And this is a paper again that we published this year that looked at that very issue. So this is sort of a summary of some of the things that we came up with as being particularly important in terms of what's on the horizon. So obviously, just ignore it. The insecticide, that doesn't mean very much. But the products, the number of products, which are a measure of how much pesticide is going in, is obviously critically important. And the more you get, the more it accounts for. So what you're looking for is you're looking for these factors that sit above this line of 0 here. These are all important predictors. It turns out that phagy, in other words, the number of plants that an insect or a mite attacks, is also critically important. So when you've got generalist species that attack a whole lot of plants, they're exposed to a whole lot of toxins, so that's also going to potentially affect their genome, which will in turn affect their impacts in terms of being able to evolve resistance. So fungi, the number of different types of plants that these attack was also a critically important factor. And the other thing that's very important is volatilism, and that's the number of generations you have here. So if a pest species has a large number of generations per year, then it can evolve resistance more quickly because the number of mutations that produce resistance is going to increase. So the more generations, the more likely you are to actually become resistant to something. So these are quite useful predictors in terms of thinking forward. So what we then did was to come up with a chart that looks a bit like this. So this is really a summary of a huge amount of work. So this is basically the situation for the top 10 pests in Australia. And obviously some of those are grains pests, some of them are not so important in grains. But nevertheless, it starts to give you a picture of risk. So risk can go all the way from, very light blue to red. So this is low risk, this is high risk. And we've also got the status on there of these particular pests. So basically, when you get a solid, you've already got some resistance happening. And when you've got an open circle, you've still got some susceptibility happening. So where you've got a situation where you've got a red circle and it's still open, that's a prime situation that you're facing in the future where risk is going to undoubtedly develop. So, so these are useful indicators for what we're going to get in the future. And it also is obviously based on a lot of data that we've collected in the past in terms of resistance that's evolved in Australia. And of course, you've got your MOH down the bottom here. So we have a few that are obviously going to be important in the future that we've got to watch out for. You can see some of the circles here still sitting there open, and they're high risk. They're probably going to get in the future, and we're going to prepare for it now. We've just got to accept the fact that they're going to come. Whereas obviously some of the other ones, light blue ones, we probably have to be less worried about. So this is sort of a way of thinking into the future in terms of what's on the horizon. And I think, I think this is data that we've, it took us a long time to get to this stage. A lot of thinking has gone into this. I think we've got to the stage where at least we can say something useful about this now, which is very exciting. So of course, once you get resistance, then of course you've got to worry about what you can do to reduce it. So I mentioned already that you've got these, you've got these resistance management programs that you can set up. But of course, given the complexity of farming systems, given what advisors have to deal with in terms of different pressures, it's not an easy ask, right, as we heard this morning. So that's quite challenging in terms of implementing those. So the question is, can we at least predict if we keep doing something, how likely it is to actually occur? And this is quite a complicated process because, of course, these pests are complicated beasts. So you can set up models, and people have been producing these sorts of models for years and years, and I think the models have become more complicated and better over the last few years, but they're still models, and of course you still need to make a lot of assumptions in coming up with those sorts of models. So here's a model that we published this year, and this is work that Virgil did out of Cesar, and of course, a bunch of other people were involved in this, and this is in fall armyworm, Spotoptera. So this is obviously a big looming pest that's becoming very important, and it's one we're going to hear about more in the next few years. And it's also a pest that unfortunately has quite a lot of resistance attached to it. So the question is, there's only a few chemicals that are left to control this thing now, and can you do something about predicting the actual likelihood of that spreading? Can you slow it down? Can you do something that's actually quite different? So these are the sorts of things that these models are good for. So the models are complicated, right? Because, insects are complicated. They spread, and, they operate in a landscape, a different landscape, some cropping, some obviously pasture, some sort of remnant areas, all those sorts of things can contain these sorts of pests. And these are pests that are often quite general. They can attack a lot of different plants. So that's the challenge that you face. And of course, it depends on external conditions. So you've got temperature. When temperature is high, then the volatilism basically, you get more generations per year. The volatilism is affected, and as a consequence, you get more generations, and that's critically important in terms of how you evolve resistance, how quickly you evolve resistance. So all these things sort of are important and come into it. And you've also got this dispersal. And you think about that and you think, oh gosh, it's terribly complicated, but at least it starts to look realistic, right? At least it means that we have a realistic picture of what we think this particular pest looks like. And there's a lot of background information, both local and overseas, that goes into these sorts of models in terms of understanding them. And then of course you have to think about the landscape. You have to model a landscape. And you don't have a uniform landscape. And these days, when you run these sorts of simulations, you accept the fact that it's a complicated landscape. Sometimes you get a patch of cropping, sometimes a patch of something else, sometimes a remnant, sometimes etc., with different suitability for obviously these fall armyworms. So this is one of the complexities that you deal with in this sort of situation. But you can simulate these sorts of things spatially, and again, that's something that's really improved in the last few years, these sorts of simulation models. So Virgil did this, and we considered 3 treatments in this particular paper, we considered a couple of chemicals, indoxacarb, and this one that I always hate pronouncing, chlorantraniliprole, terrible name. But in any case, these are the 2 that are still left in terms of controlling its pests, right? So these are ones that you want to keep in the system as long as you can. So of course, what you can do is you can put those on in different ways. So you can obviously, you can take this sort of windowing approach, or you can basically, where you put 2 out and then you put the 2 other ones out, or you can basically alternate them, so one after the other, or of course you can basically just whack them both on in the hope that you get very strong control. So these are the strategies you can think about in terms of trying to think about a realistic model that might help growers think about how you control this sort of pests. So that's the sort of issue that you deal with. And so basically you then move forward, you try and make some predictions, you see how they work, and this is what sort of comes out of all of this. So here's our 3 strategies, windowing, subsequent and simultaneous applications of those pesticides. And the first 3 factors, so the bigger the bar you see, basically, the more it contributes. And here we've got a situation where this is the probability of getting resistant to both of those chemicals. Here's resistance to one of these, and here's resistance to the indoxacarb. So you can see you've got 4 different options. This is obviously acceptable. And you can say, well, what's the probability of keeping those sorts of genotypes in the population? And obviously what you want to do is you want to try and keep the susceptibles. But you can see what happens here is that it very much depends on a whole lot of parameters. So the bigger the bars, the more important these parameters are. So these first 3 parameters just relate to the mutations and moving around the landscape. And they're obviously they're the ones that are really affecting these probabilities a lot. So if you've got mutations for resistance moving around the landscape, they can interchange, then you're going to get resistance much more quickly in this situation. And unfortunately, it doesn't really matter whether you're taking one of these 3 strategies, right? You can see that these bars pile up on top of one another very, very quickly. So it doesn't really matter which strategy you're using, you're going to get resistance if the mutations start moving around in your landscape. That seems to be the key factor. The rate is basically the amount of pesticide you're putting on. And obviously that is a key parameter. It's usually considered to be extremely important. It's probably not as important as we normally think. Sorry, I'll just rephrase that. That's the one here. The rate, where is the rate? The rate's in here somewhere. Oh, here it is, the amount of indoxacarb that's being applied and the amount of the other guy that's being applied. So these are not as important as you think. The other one that's critically important in terms of evolving resistance in this particular pest is the degradation rate. So in other words, if the pesticide is degrading quickly, you're going to get less resistant than if actually slowly degrading. Critically important. So how do we control it? How do we actually reduce that sort of thing? So it's about mutations moving in the landscape, and it's about the degradation rate of the actual pesticide that seems to be critically important in terms of slowing the resistance to these two compounds in this very, very nasty pest. So, so the question really, of course, is that's a research outcome. So how do you actually implement that? And that's the problem, right? But we can at least give you an idea about the fact that these 3 strategies seem to be surprisingly similar in their outcomes in this particular situation. So the model indicates that, it doesn't really matter what you do, you're going to get resistance if the mutations arise, but if you can somehow affect the degradation rate, that's potentially going to be quite important. So then, of course, we have the situation that, if it happens, how do we reduce its impact and spread? And of course, at what stage do you basically triage? Do you say, look, we've had a go, but we can't do too much about it, let's just move on. And of course, the key here really is monitoring, right? If you can actually monitor it quickly, accurately, you can potentially slow down this sort of process. So, so we spent quite a lot of time in AGPIP looking at better ways to monitor and developing some strategies to look at this sort of thing, and I'll talk about that now. So I'll illustrate this with respect to the red-legged earth mite. So we heard about red-legged earth mite this morning. It's obviously an ongoing problem. It's been around for a number of years. And it's obviously an invasive species, and basically we have genes that control resistance present in the earth mites, and some of it is simple and some of it is complicated. And so just to remind you that earth mites came in from South Africa, 2017 supposedly. That's the putative date that they arrived in Western Australia. And what they've done is they've moved across the country and have obviously invaded the rest of the grain belt fairly quickly. So nasty pests, also pest of pastures, canola in particular is attacked, and all sorts of legumes, etc., etc. So basically get a lot of damage from earth mites. So earth mites have evolved resistance to chemicals. So we heard a bit of talk about neonics this morning. They're not resistant to neonics yet, but they may well evolve resistance if the neonics continue to be applied. Wouldn't surprise us as scientists that this will happen eventually. The 12A group, we haven't got resistance in that yet, but we have got resistance in these two groups, the 1Bs and the 3As. So organophosphate, pyrethroid, we have resistance. So that's happened in the last 2 decades. Pyrethroid resistance first detected, not that long ago, Western Australia, and basically it has spread. Okay, so now it's in West, South Australia, and moving across. So at some stage we'll probably get resistance everywhere across the grain belt. Organophosphate, there was a suggestion of organophosphate quite early on, but it wasn't detected at this time. It became more solid at this point in time, where you've got Western Australia popping up, and you've also now seen it not just in South Australia but also in Vic. So it again is a situation where the resistance is spreading, and we expect it to go wider. So those are our two big problems at the moment, but we have others on the horizon. So what do you do about it? As a researcher, what do you do about it? So obviously, you can keep monitoring it, and you can say, well, this is what's happening, and you can monitor it with bioassays, but we want to try and do something a bit smarter and get a quicker answer on this. So the answer normally you go to is then say, well, let's do some genomics and see what we can do. So, so we published a draft genome of red-legged earth mites. Red-legged earth mite is a unique Australian pest species, so you don't go international for this. It's obviously a big thing here. Doesn't seem to be a big thing in many other places. They have to rely on local genomics. So we published a draft genome of this species, and got a reasonable genome out of it. It wasn't perfect, but reasonable. But then we've now basically moved on, and we've now got what's called a high C genome, which basically means that every single bit of the strand of the red-legged earth mite is allocated to chromosomes. So that's something that we've managed to do fairly easily and fairly cheaply in the last few months. And and this is actually quite useful. So what we've then done is we've started applying some of these genomic tools to some of our focal populations that we had available in the freezer that we could pull out where we knew something about the resistance status. So we've got a couple of susceptible populations, one from Materna South, that's a suburb in Melbourne, and Colbinabin had a susceptible population that we used, and then we also had some resistant populations, same place, and then we had these Western Australian populations that tended to be OP resistant and also varied in pyrethroid resistance. So what we then did is you basically get the DNA from these populations, pull it out, and apply it to your draft genome and see what's actually going on. And where are the differences? Where are these genes located that control resistance? So we had a situation where we had resistant populations to OPs here. We had a situation where we had a pyrethroid susceptible resistant population over here on the right-hand side, and we've got a couple of susceptibles that I mentioned. So we've got a bit of a mix here, and we had a bit of an unknown in respect to the pyrethroids, although pyrethroid resistance is incredibly widespread in WA. So we had some candidate genes here, right? Because overseas data from a long time ago and also more recent data highlights the fact that OP resistance is associated with this particular gene here. Very complicated gene, got many different parts. The ACE gene, and of course pyrethroid resistance has been associated often with parasodium channel genes, or the para gene for short. It's one of the things we discovered about the genome immediately is that red-legged earth mite has the second most different diverse as ACE-related genes to any other invertebrate that's been sequenced. That's a fascinating situation, but that's for another day. So what you then do is you then go and you look for target site mutations first. So you go to these genes and you say, well, where do we have the mutations that we think are associated with resistance? And So here's the para. So the para situation is quite simple. Here we have these populations down the bottom here. And we have a situation where there are two pyrethroresistant populations that actually have different that have different mutations as being predominant. Here's one mutation, here's another mutation. This mutation here is common in this population here. This one here has two mutations that are actually present. So these are WA populations, pyrethroid resistance. So you've got two mutations that seem to have a big impact on pyrethroid resistance in these WA populations. So sometimes the situation can be fairly simple. And we know that these mutations can control a large fraction of resistance in this particular gene. Now, organophosphate, the situation is more complicated. So we identified three mutations that are associated with resistance, and so you can see the sensitive population is one Turner, this Melbourne population, and this one here from Colbenneman. And you can see that they don't have any of these particular mutations, right? But all these organophosphate-resistant populations have different mutations at different frequencies. So here's our three resistant mutations. This one here has this particular one. This one here has this one predominantly also, but this here. And you can see the green one has these two here. So there's all sorts of mixes of the mutations that are happening here that are contributing to this target site resistance for organophosphate. It's complicated. Pyrethroid resistance, yeah, a little bit complicated, not too bad. Organophosphate resistance, target site, it's complicated. It becomes even more complicated because, I'll just skip that, because we also discovered that we have a difference in the copy number of the ACE gene. So what an insect can do, and a mite can do, is increase the copy number of a gene that's being targeted by the insecticide resistance. When you've got more copies, you become more resistant to the actual pesticide. And you can see that in these two populations, there's a Manjamat population, the treatment population, these two WA populations, you've got more copies of the actual gene that is affecting resistance. So yeah, it's an interesting situation where you have copy number variation as well as different mutations controlling this variation. So, bit of a summary: pyrethroid resistance, we've identified a couple of para mutations. Organophosphate will identify three ACE populations, as well as copy number variation to control resistance through this genomic effort. And of course, then the question is, what else is going on? Is that all that's going on? So I think we've captured most of the pyrethroid resistance in this, but we haven't captured all the organophosphates, because what we can do is we can compare pools of alive and dead mites from the same population and say, well, is there anything else going on? What else might be contributing to the live mites in this population? So to cut a very long genomic story short, this allows us to then identify new candidate genes that might not be related to the ACE, and this is effectively what we've done. So this purple mark indicates where the ACE gene is located. Okay. You got two different populations here where you're comparing these alive and dead mixes, so resistant and non-resistant. And what you're looking for is these little open dots here. Those are the outliers. So we've got the complete genome of this particular species. You can see that there's dots popping up in different places. Okay, so there's dots popping up, these open dots that could be candidates popping up that differ between these alive and dead pools of mites. So that's the challenge that you face. So we know it's complicated, that's the point. We know there's a lot of different things going on. So there's the ACE gene. ACE gene is actually not coming out as an outlier, but you've got a whole lot of other things that are actually happening at the same time. So, so what we know therefore is that there's a whole lot of other things going on in this. And this suggests this bottom line here, this polygenic adaptation, where we've got many genes and some of these are actually of quite small effect. So organophosphate is complex. You've got different genes and we don't even know what they are yet. And this is the challenge that we face. So don't panic. I still think we have solutions for quick monitoring. We can monitor pyrethroid resistance quickly, I think, in the future. We can already do that to some extent, but it's a bit slow, but in the future we can do it much faster. But what we can also do is we can come up with what I think eventually what we call polygenic risk scores, where you pull a lot of these different mutations together to come up with the risk of this organophosphate actually happening. So this is really where we're going in the next little while. And I'll just skip over this, but just to point out that there's a lot of different mechanisms. We know the target sites are important, we know the copy number is important, but there's also other things going on that we think we're going to pick up in the future. And on top of that, there's also bacteria that live inside these mites that we think may be important for resistance, but that's a separate issue. So how do we do it? Well, the Chinese, or a lab that we collaborate in China, has been developing this sort of CASP system. CASP is well known for plant breeding, but it's a very quick and very cheap way of detecting resistance in a lot of different markers at once. And they've developed this now for mites and for insects. They're using it in thrips quite a lot at the moment, as spider mites. And this allows you to really cheaply take a sample, work out, across several genes, quite a lot of genes, and actually pick up what's going on. And of course, what you can then do is you can then develop a pattern of resistance across a lot of markers to come up with one of these polygenic scores. So that's our next step in AGPIP. We're going to try and use this technology in an Australian context, even for organophosphate resistance, to actually come up with a good, simple marker for working it out. That's where I'll leave it, and some of the people involved, apart from those that authored this paper. Thank you.
Hi, Hugh Armstrong from FMC, and sorry, we're responsible for the nicknames of some of those molecules, so blame us for that. Just wondering, in the fall armyworm model, if you introduced something like mating disruption plus the use of insecticides, what would that do in terms of the development of resistance?
Yes, I mean, I think it's an interesting question. I can't answer it at the moment, but I think the model is such that you can actually add it, right? That's the point. So once you've got this basic model that works, that makes some predictions that are quite useful, you can then start adding other things to it to see what impact they actually have. So yeah, mating disruption, if it can be shown to work. I mean, the advantage of mating disruption in these sorts of models is that it's spatially explicit, right? You've got a spatial model. So of course, mating disruption is basically dependent on the area that gets disrupted. And that's why these spatial models are particularly suitable for combining those sorts of approaches. And I think that's a really interesting question for the future. But yeah, you whack it in the model, you see what impact it has. That's the point, isn't it? Yeah.
AHRI, Jason Sabini from Syngenta. Probably more a comment. There are insecticide modes of action that we could be introducing to broadacre to help manage some of this resistance. It's not a silver bullet, of course, but it's a tool. The challenge we face is that growers and advisors seem to want to gravitate towards the older, cheaper chemistry, OP and SP mainly, and there's not a lot of incentive to bring some of these new modes of action to market because diphenthioron, a good example, not new, a different mode of action, not cheap, but just not adopted, even though it's been registered and sitting there. Yeah. It's sort of sitting on the shelf and not being used. So that's a bit of a challenge, how we educate people, and I guess they won't move unless they've worn enough pain, and we've seen that in weeds as well.
Yeah, and
Any thoughts on how we try and manage that or look at other tools?
Yeah, I think that's a great question or a great comment, and I think Sam mentioned that this morning as well, right? I mean, basically, people don't do anything until it's a last resort, right? That's the issue. And when things start falling over, that's when you do something, and then, of course, it's too late. It's called the tragedy of the commons, isn't it? Based on a paper that was published in 1960, as I recall, right? And, we've always faced these sorts of issues, right? But the question is, as you say, is it simply education? Is it simply trying to do something else? Is it simply trying to get incentives going that will actually help that along? So yeah, it's a great product. There's no resistance. As you say, it's sitting in the shelf. It's a bit more expensive. What do you do about it? And if you don't do something about it, of course, then you're going to have a situation where, red-legged earth mites may be impossible to control in the next little while. Yeah.
All right. Thank you very much. Thanks, AHRI. Appreciate that.